Hello, my name is Mr. Napoltz, and this is one of my first lectures I'm going to be giving. I'll be recording my voice while also drawing out what I'm talking about, so it's more of a visual guide for those of you who are more visual than hearers. All right, so our first lesson that I'm going to be teaching is on DNA transcription and RNA processing. All right. We all know that DNA codes for proteins in our cells. If you don't know that by now, well, now you know. DNA codes for proteins in our cells, and proteins are what do the things in our cells. They are the things that get everything done. It is a protein or form of a protein that does most, if not all, of the jobs within the cell. So DNA, genes, code for those proteins. But how do we get from a coding from a DNA into an actual functioning protein? What's the process there? First, we need to start with an individual gene. Then we'll transcribe a piece of messenger RNA, which will then be processed and transported to a ribosome, where tRNA and rRNA will piece together the protein based on the messenger RNA. But this lecture, we're going to focus on the first step, which is transcription. First, let's look at the DNA sequence that codes for the protein, the gene. DNA, as we know, is comprised of two strands that are connected together by hydrogen bonds between nucleotides, which stick out of each strand like sides of a zipper. Each nucleotide is bonded to another specific nucleotide. Arginine bonds with thymine, and guanine bonds with cytosine. When these nucleotides are properly bonded, you create the double helix shape that we know very well. The first step, like I said, is transcription, which is the copying of DNA sequence with messenger RNA. All right, copying the DNA sequence into a messenger RNA. This RNA is what leaves the nucleus and transport the code for the protein. It's the messenger. So that should be easy to remember. First, though, to make the DNA code readable, you have to split the DNA strands. All right, so we're going to split transcription into three steps: initiation, elongation, and termination. All right, so the first step, initiation. The initiation of transcription has to begin with a promoter sequence, which is a special DNA sequence where an enzyme called RNA polymerase binds. This RNA polymerase will be the catalyst to create the mRNA sequence based on the DNA coding. When the RNA polymerase attaches to the promoter, it is notified by the promoter, one, where to start transcription, and two, which of the two DNA strands to transcribe. All right, you can't transcribe both. You do one or the other. So this promoter sequence helps them figure out which strand are they doing. Side note, the DNA strand that ends up being used to create the mRNA sequence is called the template strand, just like how a blank template would be used as a platform to create something new. The DNA strand that is not used is called the complementary strand. Once the RNA polymerase has attached to the promoter, there is a part of the promoter that is called the transcription initiation site. This is the site where transcription of the mRNA starts. It begins, which leads us into step 2, which is elongation, the elongation of the mRNA. All right, similar to how there is a process in DNA replication called the elongation. All right, in this step, RNA polymerase begins this process by unwinding the DNA around 13 base pairs at a time, and it reads the template strand in the 3' to 5' direction. Each new piece of mRNA that is laid down will be a nucleotide that pairs up with the DNA template nucleotide sequence. However, RNA sequencing uses the nucleotide base called uracil instead of thymine. So thymine does not exist in RNA. It is uracil in RNA. All right? Since the template strand is being read in the 3' to 5' direction, the new mRNA sequence is being created in the 5' to 3' direction, which, you may remember, is the same as in DNA replication. Side note, if the base pairing of the mRNA is complementary to the template DNA strand, then it stands to reason that the complementary DNA strand is an exact replica of the mRNA strand, except the thymines will be replaced with uracils. So when you are shown a picture of a complementary DNA strand, just know that that is, by and large, the mRNA sequence. Just replace the thymines with uracils. They may try to trick you up on a certain question in the AP test. All right, continuing on. During the elongation process, the DNA strands will rewind back together after the RNA polymerase passes them by. And the mRNA sequence will be single-stranded. It'll be a single-stranded molecule. This goes into the third step, termination. So we've started the process, initiation, we've elongated it. Now we have copied the DNA into mRNA, and we need to stop it now. So how do we stop it? Well, it's pretty similar to how it starts. All right, there are termination base sequences in the DNA code that tells the RNA polymerase to stop and detach. However, it's a lot more complicated than that because there are many different ways that this can occur. But we're not going to go into that right now. For now, just know that there are termination sequences in the DNA that signal transcription to stop, just like how there are promoter sequences that help begin the process. All right, so that's transcription in a nutshell. I'm going to be posting more videos about the rest of this process. RNA processing is next because the mRNA that we've just created and now we've detached, that is actually not what is going to be translated. We still have to take it through some processing because there are some sequences that, although they may have information in them that is valuable, they are not needed for this particular sequence. They are not needed for this particular protein. So there is a process of modifying it before it exits the nucleus so that when it goes to the ribosomes and creates the proteins, you will have a more succinct mRNA sequence. All right? Thank you for listening, and study up.